Hello, welcome to the Bioinformatics workshop. In an earlier workshop, I told you about DNA in humans and how, if taken out of one cell, the amount of DNA would be 2 meters long. I also said that that 2 meters is divided up into 23 pairs of chromosomes. If we could look at that a little bit more closely, I said that the DNA is very much like the structure of a ladder with sides and rungs across the middle. The rungs are represented by 4 nucleotides, chemicals. They're A, C, G, and T. And here you'll notice that T is always opposite A and G is always opposite C. This is the basis of the genetic code, and this is what Watson and Crick got their Nobel Prize for many years ago. More recently, scientists decided to take another look at what's going on here. They wanted to unravel the DNA, as it were, and have a look at the sequence of these nucleotides from nucleotide 1 at the start on chromosome 1 right through to the last nucleotide on the 23rd chromosome pair. Now, this is a massive endeavor. It was going to take about 15 years, but in fact they got finished ahead of time due to a very interesting collaboration or maybe competition between academic researchers and private commercial enterprises. When they started, let's say they started like this with T, they only have to read one strand because the other strand is always a complement, and they started going down there. And by the time they finished, they had sequenced about 3.3 billion nucleotides. A, G, C, T, T, G, A, C, C, G, C, and so on. It's a massive undertaking. It's hard to imagine 3.3 billion nucleotides, so let's look at it like this. If one actually typed out the letters on an A4 page in 12-point capitals without any punctuation and just kept going from the very first one to the 3.3 billionth one, put them on pages like this, bound the pages together in something like telephone directories, and stacked telephone directory after telephone directory on top of each other like this, in the end, the stack of telephone directories would be 15 floors high. Now that's just the genetic information that one gets from half of the chromosomes. That's the set from mother, for example, 15 floors high. There will be a similar 15-floor high stack of nucleotide sequence for father. This mammoth endeavor was called the Human Genome Project. The word genome means the total amount of genetic information in a human, for example. The information is obviously not stored in phone books like this. It's stored in extremely powerful computers. And this means that any scientist working on any DNA sequence anywhere in the world can dip into this database, the Human Genome Project database, and extract from it the information that they need for their work. At the same time, quite a few other organisms were given honorary human status, so that many hundreds of other organisms have also been sequenced, from viruses through bacteria, plants, and animals, and that information is also stored in databases. The storage of all this information from all the different organisms in very powerful databases has spawned the study called bioinformatics. Bioinformatics is about using computer-based data in biological, medical, agricultural applications. And we'll talk about that a little bit more in a moment. One of the first very interesting things to come out of the Human Genome Project was how many genes there are in humans, because the genes are strung out along this 2 meters of DNA like beads on a string. And we had always wondered, how many genes are there that make us humans? What I mentioned is that as well as human DNA being sequenced, other organisms were sequenced. For example, mouse. So let's pretend that this wire represents the genome of the mouse. It's much easier to do biological experiments in mice. You can do things you can't do in humans. And so scientists were able to predict that mice had about 40,000 genes strung out along their chromosomes. If you like, you can think about it like a recipe. Previously, I likened our genes to recipes. And so bioinformatics is as if we've got a lot of different recipe books. And each recipe book is for a different organism. So now we've got 40,000 in mice. How many do you think we've got in humans? Can I have some guesses from the students here? Yes, you'd think so, wouldn't you? A lot of people think we're going to have a lot more than mice. And one of the most surprising things originally that came out of the completion of the Human Genome Project was they could actually only find about 27,000 to 30,000 genes in humans, which would mean it looks as though we've got less genes than a mouse. As more research is being done, others are being found. But the studies emphasize the amazing complexity of the human genome. And the years of research still to be done in the future will give us more answers about this. So all the different gene numbers are also kept in catalogues that can be queried by anyone who's interested in this aspect of science. The Human Genome Project has gone way beyond looking at the numbers of genes along here. It's looking at the products of the genes, which we call proteins, and it's going on into functional studies where we look further to understand how the different proteins make us the way we are, the mouse the way it is. Bioinformatics, as I've said, is an extremely powerful tool that has innumerable applications in biotechnology. It's used in biomedicine, agriculture, genetics, and many other sciences. Let's look at it for a moment in forensics, which is what we talked about in one of the earlier sessions. The small differences that we have, one from the other, looking at the group around here, are the result of small differences in our DNA sequence, and the markers that have been developed that are used in forensic testing are actually based on little strings of DNA sequence. So what I'm representing here are just individual nucleotides. And you can see that here we've got two nucleotides that keep repeating one after the other, CA, 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 CA. And the number of repeats on one person's chromosome may be different from the number of repeats on another person's chromosome or on the chromosome from their other parent. So here are some different examples. And the science of knowing this allows the forensic experts to decide which particular areas they will look at to find differences between people. The information that I've talked about being stacked in those 15 stories of telephone books is actually stored on computers. At this stage, we're not right there for the technology, but it is predicted that within the next few years, any human will be able to get their total genome sequenced and put onto a special kind of CD, which they will take with them whenever they go to their doctor, can be read in the doctor's rooms, and will give background information about their genome and the underlying conditions that they are susceptible to. It's also used in agriculture, and it's used in making genetically modified organisms. When we were talking about genetically modified organisms, we decided to look for a gene that would confer drought resistance on a maize plant. I said, how will we find that gene? How will we know if this is the genome of the cactus, where the gene we want lies? And the way we do it is to refer to the cookbooks that I was talking about, looking for the recipes. So if this is the recipe book for one particular sort of plant that has already been studied and its information put into the database and analyzed, we'll be able to go and look for its recipe that confers drought resistance. Now we can search through the sequence and the areas of our new plant, a cactus, and look for a similar gene here, and that's how we know to cut it out. This is a powerful technique known as comparative genomics. We've come in a tremendous way in the last 10 years, since the completion of the draft sequence of the human genome. We've found out things we'd never even dreamt of, like the number of human genes, for example. It's very hard to predict what we're going to find out in the next 10 years, but we can be pretty sure that we're pushing back the boundaries of science with the technology of bioinformatics.